Okay, we'll pick up after our 10-minute lecture on Wednesday. What we're going to be talking about is sort of an overview of growth, then cell types, then we'll start at the outside of a cell and work our way in over the next couple of lectures. And just to give you a setup, and I think I have a graph of this information in here. Used to have tile in here, and so I was able to actually count off the size of the room. So if this were a plant cell, mitochondria would be roughly the size of a human. Chloroplasts would be roughly 2 to 4 times larger than a human. A nucleus would be about 12 feet across. And depending upon the cell, if this were a mature cell, the rest of this would be water in the vacuole. And if you blow the cell up to this size, and again, I'm going to go over this again, the cell membrane would be thinner than a piece of saran wrap, which tells you how strong a cell membrane can be. It's even if you blow up a cell to be this size of this room, you'd barely be able to see the cell membrane. Okay? Now, we finished up yesterday before the thing stopped recording, talking about meristems, apical meristem, and that plants have these other meristems. So if we go in and cut off this top here, then these meristems then become active and start dividing and producing cells. Yes, ma'am. I thought I did. Thanks. Thanks for reminding me that. I hit this one, which apparently, I guess, is it coming? It's coming out now. Okay, give it a couple seconds. Okay. So we can is it going off again? Oh, there we go. Okay. So we can look at these then. So all the cell division occurs right here. And then if we then decapitate this thing, these will start growing. Okay? And they become then new apical meristems. And so what you want to think or get out of this class is things are three-dimensional. So this is coming behind and in front of the screen. So you have, if you look at and I'm sure everybody's looked at the apical part of a plant, you'll see it's got just a tremendous number of little leaves around it. And in those little leaves are little meristems. And most of the growth occurs by elongation. And you have this sort of zone of elongation where the cells increase their length. Again, they're growing up and down. Plants are polar. They grow up and down by thousands of times. So by the time a corn plant is, like I said, it's the only really plant I know anything about, is about this tall, almost all the divisions have already occurred. And you can take that corn plant apart and you can see really, really small ears. You see really, really small tassels. So almost all the growth in a plant is due to elongation. And we're going to talk a lot about elongation. We'll talk about cell walls. Then we'll talk about it again when we talk about hormonal control. Now we have the other end, which we can look at. And it's a little more difficult to see, but again, there's this. Why is it trying to move that? It's their zone right here where divisions occur. And if I go to this one, you can start seeing these cells elongate. And the root then is pushed down. And an additional structure that a root has is this area right here, which are extremely heavy-walled, very thick cells called the root cap. And what's the purpose of the root cap? Right. Yeah, to protect the meristematic zone, the root meristem. And if you think about it, if you go to a macro or a micro scale, the root is continuously going around things. It's going through the soil, but the soil is composed of particles of different sizes, of different makes or whatever. So it's continuously pushing down to separate those particles. And if it hits something like a rock, well, the root will grow sideways until it gets to the point of where it's always trying to grow down, so it'll go sideways and gets to the side of the rock, then it'll go down. In other species, if a root encounters something like that, the root growth will slow a lot and other roots will start being produced. Because the roots are where minerals and water enter the plant. Okay, questions about that? Okay, now we're going to start talking a little bit about cells and cell types. And then I'll go over this a little bit, then we'll go over it again when we talk about each individual organelles. And basically what we're looking at here is a cell inside a wooden box. And so if you want to visualize what's going on in a typical plant cell, is you have this membrane enclosed region, the cell proper, inside the wooden box, and continuously you're standing there and you're pumping water into the cell. There's always, if the plant has enough water and it's not wilted or anything, the plant continuously pumps water into the cell, which causes the cell to swell up and push against the cell wall. And as it pushes against the cell wall, and when we talk about water potential, we'll go talk about this in detail, water goes out. So water molecule goes in, water molecule goes out. So that is the typical thing you see when you see a weed out here. Now, if it's in the middle of the summer and the plant doesn't have enough water, there's not water being pushed against the cell, into the cell. So the cell wall, there's no tension, there's no force built up on the cell wall, so the plant wilts. So when you see a wilted plant, you're looking at a plant that does not at that time have water being pushed into each individual cell. Okay? So, make it a little more complicated. If we look at most mature cells, we have this huge vacuole. So you have a balloon full of water inside a balloon, and then that is inside a wooden box. And you can think of a primary cell wall as being like a burlap bag, which actually looks like a burlap bag. And you put that balloon in there and start pumping water into it, the balloon's going to swell up. It's going to completely fill the volume, so you're going to get whatever the shape of the burlap bag is. And when growth occurs, those pieces of burlap then will start coming apart, and the water pressure will push them up so the plant will grow. The cell will grow. Okay? And we'll go over that again. A few things that plants have. They have a chloroplast, which probably is the most unique thing in plants. Otherwise, they're pretty similar to animal cells. And of course, in a mature cell, we have this large vacuole. Okay? Now, something that you usually don't get is the fact that you get this notion that you've got this cell wall that surrounds a cell, but at any given time, there are actually openings between cells. They're actually like you have a wall here, and I actually cut a hole in it to the outside, to the next wooden box, and I cut it through there. So it's not unusual for cells to have these direct ways of moving material from one cell to another without crossing a cell wall or a cell membrane. And on the downside, that is the way some viruses may move. They move not through the cell membrane or cell wall, they just move through the holes. And you'll see some EM of these plasmodesmata, which they're called. And there's different kinds. This one's called a simple pit. It's just an opening. They can be very elaborate. You're going to see some later on. So we have this primary cell wall, and then when the cell gets to be what it's supposed to be, when it gets to the size that it should be, then it starts producing secondary cell wall material. Okay, and that's always inside the primary cell wall. And I like this because it shows you all these potential layers of secondary cell wall that can occur. And a lot of times, in addition to just cell wall material, we have this compound called lignin, and we'll go over that again. And lignin is what makes wood. Okay? Lignin is like cement. You can think of like a primary cell wall as being a burlap bag, and then take that same burlap bag now and coat it, impregnate with cement. Okay? That burlap bag is not going to move anymore. And you can think of the lignin and other things that are parts of cell walls as strengthening that. That's why trees stand up. It's because they have that tremendous amount of lignin in the wood. That's what wood is. And we still don't know everything about lignin. A lot of work going on to understand lignin. Why would that be? A lot of money going in to understand how lignin is actually made, how it actually is incorporated into tissue. Who'd be paying money for that kind of research? People that build houses, maybe? If we know exactly how wood is made, then we can start thinking about modifying it. So actually change the characteristics of wood. If I go to an extreme, if I'm a paper company, I would love to have wood without lignin in it at all, right? Because paper is not made out of lignin, it's made out of cellulose. So paper companies use soft woods so they can easily get rid of the lignin, but what they'd really like to do is to come up with just cellulose, or as much cellulose and as little lignin as possible. Go to the other extremes. If you're building a house, there are going to be certain kinds of wood you use because they're strong, and they're just like steel. So the kind of trees you would use for building would be different. Okay? Now, let me go back here just one point. This is sort of a diagram of what these secondary cell walls look like. Here's two kinds of tissues that are very important. You've probably heard of them before. On the left, it's called xylem, and on the right is phloem. And I've got a couple pictures of these I'll show you in just a second. But there are different kinds. You just need to know that there are tracheids and vessel elements for the xylem. And if you look at these things, look at these incredible amounts of secondary thickenings they have. What does xylem do in the plant? Transports water and minerals, and that's it. And it also goes from the bottom to the top. Okay? And at maturity, trachid and vessel elements are dead. There's nothing but an empty hole and these elaborate end caps that you'll see. So what's the purpose if we look at, let's look at these two right here. You've got this, it looks just like a spring, and here you've got more or less circles. What's the purpose of these secondary thickenings? What does it do? Why are they there? I'll ask this question a dozen times and for totally different things, and it's always the same answer. Partial structure support, but the big thing is to increase surface area. Increase surface area so water can hydrogen bond. And when we talk about water potential, and we talk about how it's impossible for water to get into really tall trees, but we know it does, has a lot to do with the structures of these xylem, tracheids, and vessel elements. They are dead. They are open. They form continuous pipes from the bottom to the top of the plant. And again, like I told you a couple lectures ago, it's a conundrum. The only way plants can take up water and minerals is to lose the water out of the leaves. Yeah, sort of a crazy thing to have evolved. But it's the only way that can explain, it's the only way that water can move into tall things, okay? Now on the right we have a sieve element and a companion cell, and that's phloem. And I've got a diagram of these two later, or a picture that's a little bit better. What does phloem do? Sugars, organic compounds, up and down. Okay? And what's really interesting is that these tissues, these sieve cells, and these sieve tube elements are live. They have membranes. And what's more interesting is these large, long sieve cells do not have nuclei. And they count on the companion cell, and here's a shot of the nucleus here, to provide the information to produce organelles, enzymes, this kind of stuff that's required for the phloem to work. And you know, if you look at a tree, that 99% of what you're seeing is xylem. And the phloem is that little tiny ring around the plant, you know, and that's where maple sugar comes from, of course, as you're cutting into the phloem and you're harvesting the sucrose and the flavor molecules that makes maple sugar taste like maple sugar. And you also know if you girdle the plant, if you cut continuously through all the phloem, you've got a dead plant after a period of time because it doesn't have any way of moving nutrition around. You can still move water, but it can't move organics. It can't move sugars, amino acids, hormones. A lot of hormones, as you will see, moves through the phloem. Okay, questions about that? And when you look at phloem, it's really, really hard to see the companion cells. They're very small. And they also have these things I was telling you about called plasmodesmata, so things can easily move from this companion cell with the nucleus into the sieve cell. Okay?
When that plant's growing and those cells are dividing, every time they replicate, do they have attached from creation that companion cell with that nucleus?
That's a good question.
Or is that the nucleus that tells it to replicate?
So when they're growing, what happens? Okay, when they're first growing, they're not differentiated. Okay, they're just cells. And you may have heard of a cell called a parenchyma cell. That's sort of like a basic cell type. And then as the cells, then they start elongating, and where they are in the plant will determine what they become. Okay? And we're learning that's even true with animal cells. That they've had a lot of success lately with printing organs. And it turns out if you make a scaffolding out of a certain kind of material, let's say, and they've done this to some degree with liver, and then you spray these cells on that just the shape for some reason determines what that's going to, so it'll start becoming liver cells. It'll start producing vascular tissue. With plants, we've known this for a much longer time. Where the cell is in the plant, as it starts to mature, due to hormones, the really intricate interactions of all these hormones and location, then they will start developing. Okay, so then as they become more mature, and the question is for every sieve element, is there a companion cell? And the answer is sometimes and sometimes not. So sometimes you will see companion cells that are really large in comparison, that are much larger, but in most instances you can't identify one companion cell with one sieve tube element. Okay? And it's actually hard to see the companion cells. They're so small. As opposed to xylem, which is 100 times bigger, more or less. So whatever differentiation, and we'll talk about differentiation, we'll talk about hormones. You know, at some point, because of the environment, the hormone environment, whatever else, that cell then becomes fated to be a xylem or a phloem element. Okay? And you know also that, and people do this a lot with mint because you can see it easily in mint. If you, mint's got a square stem and the vascular tissue's in the corners, so you can cut a corner out, and then the plant will start producing parenchyma cells to fill up that hole. And then after a period of time, those parenchyma cells will differentiate into replacement xylem and phloem elements and reestablish transport. Doesn't happen in all plants. Okay? So here's a cross section, and just to make the point here is here's the vessel element. Okay, these are things called fibers. Fibers are just incredibly thick parts of the xylem that are basically for support. And right here, a little bit here, and a little bit here, that right there, that little tiny bit there is a companion cell. Okay, and this is phloem. So basically, if you're looking at a cross-section, you identify the xylem, and since you can easily identify the xylem, the stuff close to it is probably going to be phloem. And then you can stain for nuclei, you can stain different stains. So a lot of what you see is the process of elimination. And every plant species looks different. You know, I've had 24 course hours in microscopy, mostly electron microscopy. And the way you identify things is a lot of time, it's got to be this because it can't be this. So chloroplasts are neat. Everybody knows what a chloroplast looks like. It's hard to find a companion cell unless you do that for a living. You know, and I've actually sent some micrographs to this guy at the UT when we were working on phloem, because I wasn't sure I was looking at phloem. Now, if we look at corn, here this is a color, a stain that stains xylem elements red, and you can see them popping out here. And then if we look at another cross section, you're going to see the larger xylem pieces that you're going to see here. Mainly because in terms of amount, the plant needs a lot more water than it needs sugar. So it can fill its needs for carbohydrates with a much smaller amount of tissue. Okay. And then you can look up into this area, and you can sort of see here, and this would be some of the phloem. Okay. And I want to sum up with what the major functions are of these two tissue types, because you're going to see them again and again and again. A xylem moves water and minerals up the plant, and may move some minerals down. That may be possible too. But primarily it's moving water from the base to the top of the plant, and it just carries the minerals with it. There's no transport. You've got a hollow pipe from the bottom of the plant to the top of the plant. And you probably know that some herbicides or some these compounds, insecticides, can be transported through the xylem so you can get it inside the plant. Now, on the other side, the phloem, it's much more complex. There's, of course, water in there, but there's also food, sucrose, other things. And in this case, the materials must be transported across the cell membrane. That's a big deal. Okay? There can be some plasmodesmata, but a lot of things have to be grabbed by a protein and taken across the membrane to get to the next cell. Hormones are usually grabbed. Actually, most hormones never even enter the cell where they produce their action. They bind to the cell membrane and release factors inside the cell. And about 2/3 of the way through, we'll be talking about how hormones work. So if you're moving across the membrane, it's got to be living, highly metabolically active. Got to produce a lot of ATP because most all movement involves ATP. Very complex. Okay? Questions? Okay, let me go to the next one then. Why is that not? Okay, now we're going to be going from the outside in. So what we're going to do today is spend a little bit on cell walls, and then talk about cell membranes. Plant cell, basic unit of the plant. Like I said, a balloon, you can visualize it as a balloon inside a cotton woven bag, and then at maturity that becomes hard. It becomes wood. So it goes from sort of a primary cell wall, you can envision it's, like I said, a burlap bag, or a bag made out of, cotton fibers. Much more complex than that. And so we're going to be talking about the stuff that is outside the cell membrane. So if we look at the first structure, we have a cell wall, a middle lamella, and then the next cell wall. And the middle lamella, or middle lamella, depending on how you want to pronounce it, helps stick the cells together, so it's sort of sticky. When cells are first produced, they, of course, only have primary cell wall material, and you will hear them called ground tissue are parenchyma cells. And in the plant world, a parenchyma cell is basically an undifferentiated cell. It can change into a lot of different variety of cells. So like I said, if you take that mint plant and you take a razor blade and cut out a little bit of the corner, the first thing that's going to be produced there is parenchyma cells, and then those parenchyma cells will differentiate then into xylem and phloem and whatever else is there, okay? Now, this is a couple of pictures. You probably did this on the left when you took freshman biology. What you're seeing there is probably an Elodea leaf that's had some sucrose water added to it, and that causes the plasma membrane and everything inside to shrink, okay? So what you're seeing in the center there on the left-hand side is the protoplast. The protoplast is the living portion of the cell. Cell membrane inside is called the protoplast. And if you did this experiment, you're seeing the cell wall, you're seeing the living part of the cell is now pulled back. And you can actually take this piece of tissue and you can spray it, you can remove the sucrose water and add water to it, and the protoplast will refill the wooden box, okay? So when you see this, you're looking at a plant that, if this were a leaf, would be wilted because there's no pressure. The protoplast is not applying any pressure to the cell wall. Does that make sense? And then the right is something we can do and have been able to do a long time, is a cell wall where the protoplast, where the cell wall has been digested away. And this has been going on since the 70s. So you can take tissue, you can add cellulases and other enzymes, and you can break down the cell wall, and you have a living cell, and this living cell will regenerate a cell wall. This is sometimes used in molecular biology to move things into the cell. There are other ways of doing it now. When this first came about, people had these wild ideas that you could take protoplasts and fuse them together. So you could take a soybean protoplast, infuse it with a corn protoplast, and produce this super plant that would, have the protein complement of soybean and also the protein complement of corn. Well, that, of course, doesn't work. What you would get was not much. But this was before molecular biology. This is the way people were going to improve crops. So you could have a corn plant that fixes nitrogen. Unfortunately, nothing like that. You could even fuse human and plant cells together. They never did anything, at least not so far. But you can add things to them. If you're doing research, you can add things to them that may not be able to get across the plant cell wall. And so you can see if something's expressed in the protoplast. That is used a lot for people that are looking at gene expression. Okay? Now, got a couple just of word slides to talk about what the cell wall material really is. And you think of cellulose as being cell wall. Well, the majority of cell wall is not cellulose. So if you think of cotton, which is cellulose, then what you will see, and I've got a picture later on, is you have this thing called a microfibrils, and they are linked together. And then think of the microfibrils being the little strands or the burlap in a burlap bag. Okay? And they're held together. They're very strong. They're linked by hydrogen bonding. And the bonds can be broken. And we'll talk about that in a little bit. Then we have this extracellular matrix material, which is actually most of the cell wall. And it can be several different kinds of compounds, which we'll talk about in a bit. Everything is deposited inside the primary cell wall. So the secondary cell wall material is always inside the primary cell wall. What does a cell wall do? It's pretty obvious. It keeps the protoplast from exploding. So if I isolate protoplast, I have to add a lot of sugar so that the protoplasts just don't blow up. So a protoplast inside a cell wall expands until it hits a cell wall. If you take the cell wall off, that protoplast would blow up because you're pumping water into it and it would finally just pop. Okay? And that's going to determine the shape of the cell. So the way that cell wall grows or loosens is going to determine how the cell grows. It's going to grow this way, across. If it's in a leaf, it's going to grow maybe left to right. If it's in the stem, it's going to grow up and down. The cell wall material determines how it grows. Hormones are involved, and you'll get that a couple of times. So it can expand. It also, obvious structural barrier, provides support. Not all cells have secondary cell walls. Some stay primary. Here's a really neat little picture of cell wall. When I say it looks like burlap, it really does. What you're seeing is the microfibrils. These are the cellulose fibers that are just cross-linked. And then there's material that sort of sticks them all together. So that basically looks like a burlap box. Now, what else is there? We have cellulose. We have these things called matrix polysaccharides, which are mostly pectins and hemicelluloses. Anybody ever heard of pectin? What's pectin? What do you use pectin for? Jelly. That's exactly what it does in the plant cell wall. It's a gel, and it's a gel because it's has bonding between the smaller molecules to form a gel. And that's exactly what it does in the cell wall. It basically sticks itself between the microfibrils and bonds those cellulose pieces together. Hemicelluloses are just another class of compounds that are not quite, they're not just cellulose, they have side chains. Lignin we're going to talk about a little bit, and then there are also some structural proteins, and I don't know if I added the new slides to this one or not. If we look at what we see, cellulose, and this is important, is beta-1,4 glycoside. So it's just a bunch of glucose molecules strung end to end with a beta bond. Instead of what's alpha-1,4 glucose chain? Anybody know what that is? That's starch. Cellulose and starch are exactly the same thing, except cellulose has a beta, which means every other glucose molecule is in upside down. And we can't digest cellulose. But we can, of course, digest starch. Starch is an alpha. And so what this picture is trying to show you is these individual chains, and then the blue, if you can barely see that blue, that's the bonding between these cellulose chains. In this cartoon, we're seeing how these things go together. So you have these individual chains of glucose that go in to make this fibril, this microfibril, and that's what you see in the cell wall. And glycans just have other carbohydrates added to the glucose chain. So you've got this repeating glucose, and with glycans you can have xylose, you can have other compounds that are attached to it. So that then, just like I said, then embedded into the gel, the pectins and the hemicelluloses. Okay? Why is it gel a gel? Because all those things are either covalently or hydrogen bonded to keep them from sinking to the bottom. You know, they're interacting with water, they're interacting with other molecules to form a matrix, which makes it a gel. Okay? Now we really every couple of years someone comes up with a new way of the new cartoon of what cell walls may look like. And so what you're seeing here in this cartoon is, of course, the cellulose fibrils, and then here's a xyloglucan, here's some pectin, and these things can bond to the cellulose. Okay? So they are chemically bound to the cellulose, so they don't move. If I want the cell wall to expand, I don't break the cellulose, I break those other bonds, and then the bubble or the balloon will expand. The cellulose pieces will slide by each other because the balloon inside has water going into it, so it stretches apart, and then those bonds form again, and then it becomes stationary. Okay? And I'll tell you now, this is done. The hormone that does this is auxin, and what auxin does is it ends up putting protons out into the cell wall, and those protons cause these bonds to break, and the cellulose fibers then can slide by each other. Again, go back to pumping water into the balloon inside the bag. Okay, and if I take that cellulose burlap bag and I cover it with pectin, then the pectin's going to interact with the cellulose, and it's not going to grow. It's strong then. Okay? Now, I think there's a more recent. Okay. So what's pectin? Polysaccharide. Used as a gelling agent, yeah. The main thing you need to know is pectin is basically what holds the cellulose fibrils together. Okay? And I'm going to stop with this one, and then we finish up. What is today? Friday, so Monday on cell walls. And here's a cartoon of how these things again may be arranged. What we're going to talk about next time is the cellulose synthase complexes and how the cell wall material is made, which is pretty important. And we'll first start off with blossom-end rot. Anybody know what blossom-end rot is? In tomatoes, the bottoms of the tomatoes sort of rot. One of the main things that cell walls need is calcium. And calcium causes all these other interactions to work. So if there's no calcium around, the cell walls can slide apart, So the protoplast blows up, and you're ending up then, and this happens at the blossoming of the tomatoes, and so all that tissue just blows up. Without calcium, the cell walls can't be stable. The calcium interacts with the pectin to keep it from just expanding out like crazy. And remember, if the cell walls break down, the protoplast explodes. And so what you see is a lot of dead tissue. Okay? Okay, see you Monday.